All right. I think we're all good. Welcome back. So now we're going to hear from a couple of scientists, some scientific presentations. And just to give you a little explanation first, we were hoping, I think I mentioned, or maybe I didn't in my very early introductory remarks, that our agenda's not quite as tight as we like to have it, generally sort of topically. And part of that is because we had some scheduling challenges. And one of them was that John Gensel, who will be here tomorrow, couldn't make it today, was going to be in this session with Cedric and Candace. And they actually, the three of them, do some work together, some collaboration together. And then Cedric has to leave today. So I was trying to figure out how to put them all together so they're separate. But what I just wanted to give you briefly is, if you recall from Dennis's slides, he put up a slide talking about, we heard from a lot of scientists about questions about models, animal models. And so the presentations you're going to hear from Cedric and from Candace and from John tomorrow are a little bit of an exploration around the models that scientists use to model spinal cord injury in an animal. And questions not only around how they do the injury, which is one question, you know, a clip or a contusion or a hemisection. There's all different ways scientists do that, but also these questions about age. How old is the animal? How long is the injury? Gender of the animal. These are all variables that are important. And Candace, as if you don't already know, works in a pig model. And so we wanted to have this conversation from each of them about how they model the injury and what does that mean and what are the implications of it. So please welcome Cedric Geoffroy from Texas A&M. Thanks, sir.
Thank you.
Being here.
Hi. Can you hear me? All right. Well, thank you, Matthew. Thank you for the invitation. Very happy to be here. And I realize I packed a lot of information, so I will try to speak slowly, but at the same time, speed up through the slides. So today I'm going to talk about the importance of age and the age factor after spinal cord injury. So in my lab, beside the age factor and axonal regeneration, we're also working on liver dysfunction, bowel dysfunction, and heart dysfunction. If you have any question about that later, just please come talk to me, and I will be happy to explain what we're doing in the lab. But for this presentation, we'll be only focusing on axonal regeneration and sprouting in the context of aging. And first, I want to thank Teresa Sutherland, the postdoc in my lab, and Natu Sefiani, who have been working specifically on this aging project, and several collaborators that provided us with different compounds that we'll present very briefly today. So I guess the main thing I want to talk about today is, do we have a problem with the current drug discovery process? And you most likely you've seen this kind of schematic before, when you have different stages in drug discovery, when you do all the in vitro work, you move to preclinical trial, and hopefully you have a few compounds that work in rodent or the model, and then you move along the clinical trial. And as we all know, and that's so far, it's not very working well. And just an example for Alzheimer's, but it's the same thing for spinal cord injury, where we have almost 100,000 papers published, many different compounds that have been tested in 400 clinical trials, billions of dollars invested in the clinical trial, and so far, not a single FDA approved beside recently specifically for Alzheimer's. But anyway, the point is it's the same for traumatic brain injury, spinal cord injury, etc. And today I'm going to talk about how maybe and propose some kind of suggestions and recommendation how we could try to improve this discovery process by modulating, by specifically looking at the age factor at both the drug discovery and in vitro system, as well as the preclinical system. And I believe Candace and John will talk about that today and tomorrow. So first of all, why is age factor important? For the past few decades, there was this massive increase in the average age of incidence from 29 years old in the 70s. Today is about 43 years old. Not only that, but the census from the Reef Foundation years ago clearly showed that the massive distribution differences depending on age, and about almost 80% of the people living with spinal cord injury are 40 years old and older. And that seems, when I look in the room, that seems very representing of this graph bar here. Now, one of the major issue, and that most of the people have been working in preclinical studies, we use 2 or 3 month old rodent model, usually to test a drug or whatever you want to assess. 2 or 3 years old mouse or rat, at best, you're in that 15 to 18, like very young adults. If you want to work with a corresponding age of 20 years old in humans, we should start working in middle age, like a 6-month-old mice. If you want a middle age of 40 years old, we should go to maybe a 12-month-old. And older humans, we should go into the 18-month. So clearly there's a dichotomy between what's happening in preclinical stages and what's happening in humans. And in stroke, they found a lot of different compounds that was very effective in vitro and effective in preclinical model. When they moved in humans, pretty much everything failed. And one of the main reasons is because in stroke, it's occurring in much older population. And that would make sense that age and the way the drug was acting was not efficient in the animal model and the human model. So clearly there's a need to better understand how age can affect the central nervous system repair. So today, I'm going to reflect quickly on some in vivo work that we've done in the past in trying to understand how age can impact axonal regeneration and axonal sprouting using 2 different spinal cord injury models that I would describe. And finally, I will try to and hope to convince you that it's really important that we use adult cells in vitro before moving in preclinical animal in middle age and age mice before even considering moving to clinical trial. So for this particular age-dependent regeneration decline, we use a particular gene called P10. You don't have to know about it, but so far it's one of the most efficient way to target the cortical neurons that project all the way to the spinal cord and control your fine movement. So if we do cortical gene targeting of this P10, it's been shown many years ago that you can enhance axonal regeneration. So years ago in mice using the dorsal hemisection model at T8, where we cut the dorsal half of the spinal cord, with very basic question, can we repeat this gene targeting that we see here? We have an increase in axonal regeneration that you can look in the yellow axons going through the glial scar and caudal to the lesion. But the main question is, can we repeat that in other animals? And the answer was no. Using the same transgenic mice, just based on aging, we can clearly see there's a reduction in axonal regeneration caudal to the injury. What was interesting to us was that regeneration seems still to occur rostral to injury, and that's what we quantified here. So it doesn't matter when you do the gene targeting, you will enhance axonal regeneration rostral to the injury to the same level. So that makes us think that maybe there's something in the spinal cord injury itself with age that was altered, that was reducing axon growth. And I won't go into too much detail, but we're looking into at the astrocytic response in astrogliosis. And indeed, with age, we have increase in astrogliosis that can impact axon regeneration. I'm going fast because I told you I put too much information on my talk. So we're looking into inflammation, and same thing, we observe that with age, there's a stronger inflammation, which again can reduce axon regeneration. So right now we're trying to understand specifically what's happening at different age and different time point post-injury at the injury site. And one of the things we found is that we see some increase in fibronectin in the fibrotic scar that again can inhibit axon regeneration. We see changes in other molecules that are involved in inflammation. And so bottom line is, with age, that's a review that we published a few years ago. With age, all these kind of cell type can impact the recovery and can impact axon regeneration. So the glial scar, the myelin-associated protein, the extracellular matrix, all this molecule can inhibit axon growth. So because I wanted to understand how the molecular, within the neurons, the molecular pathways are changed, we switched to a different model called pyramidotomy to assess sprouting. And this is a different model. I don't know if Candace will talk about it, but basically we do injury on the right side of the cortical spinal tract, and we gene target the cortical neurons on this side. And then we measured the compensatory growth from uninjured axon, another way to see if we can enhance sprouting and plasticity and enhance functional recovery. So the reason we added from this P10 targeting gene to this SOX3 is because it's been shown that if you co-delete, you increase in sprouting. So usually you don't have any axons on that side of the spinal cord. If you do the gene targeting, you can appreciate this massive increase in growth. And years ago, Jigang lab in Boston shown that if you do this P10 SOX deletion in very young mice, you enhance functional recovery of falling function. And so we did exactly the same where we first did the gene targeting and then did the injury, but then we did the reverse because we wanted to see if there was some translational potential. So the second essay we did is first we did the injury and weeks later did the actual gene targeting to see if we can enhance growth. And bottom line is, if we do this gene called P10 targeting, it doesn't matter when we do the gene targeting at postnatal day 1 or 12 months, but this is the green line that we see here, we see the same increase in axon growth. What was surprising, if we do this SOX3 gene targeting, it can promote growth in young animal, but we can no longer observe axon sprouting in older animals. So clearly showing to us and to me that there's some age impact on specific pathways. And this is just two particular examples, but you can imagine that if you target a drug, you use a drug that promotes the mTOR pathway, which is downstream of this P10, it may work regardless of age, whether if you use a gene targeting or drug targeting this pathway, it may not work the same way. The other thing we found is if we delay this injury model and we do the same gene targeting, we observe the same thing with the massive increase in sprouting in the younger and the middle-aged animal, but we no longer observe the effect of the SOX3. Again, confirming that the age-dependent change in the signaling. So from this summary, and again, I went very quickly, but I hope that I've shown you that with age, there's some changes within the neurons and changes within the spinal cord injury itself that can reduce regeneration, reduce sprouting, as well as it seems to be pathway specific, at least in the response from the neurons. And I've shown you there's some different players that can also impact axon regeneration with age. And to me, this is very important for translational approaches. And that's something I just mentioned. If you target specific gene A, it doesn't mean it's going to work the same in young, middle age, or the mouse and or humans. And the same thing with the same drug that we target the same pathway, it could be age dependent. So clearly we need to understand in each cell type and different time point post injury, how are the molecular signaling changed in order to better come up with way to enhance growth, enhance regeneration. So I told you that the main idea of the talk is to try to see if we have any change, any issue with the current drug screen. And here I just want to point out 4 different ideas. So the first one is when we worked in drug discovery, we use cell lines, or we use embryonic stem cell neurons, or even induced iPS cells. But these they're like young embryonic neurons. They do not match what's happening in clinic. And that actually, I will show you some data, leads to false positive, meaning you have some drug that can enhance neuroprotection and enhance growth in vitro, but they will be unlikely to do so if you use adult neurons specifically. So that's what I call a false positive drug, because they're very unlikely that if we move them along the path to be successful, because they're only working embryonic neurons. The other negative aspect of our current strategy is we have false negative, and I will show you an example of a particular drug where it doesn't work, or actually is detrimental for embryonic neurons. It reduces growth and it reduces protection. But if you use the same drug at younger adult or even middle age, you will have increase in neuroprotection. So clearly, the way we're using the drug system right now can actually lead to false negative and potentially drug that could be move along the clinical all the way to the screen process. Another aspect that I will quickly show you in the next few slides is that we find drugs that actually may be detrimental depending on age. So the same drug, if you give that in vitro, at least to young neurons or middle-aged neurons, will promote growth or kill them. And that's a very important consideration. If you have a drug that works in preclinical in young animal, most likely if you move along the clinical trial in other animal, actually in other humans, actually it can be detrimental. And of course, if you combine all this together, all the drugs that we've been testing for the many years, I think to me, we're wasting time and we're wasting resources to some extent because we're not targeting the right cells in the drug discovery, and we're not using the right model in preclinical. So what our solution, and again, we don't have a specific proof that it will enhance all the way to clinical trial. Right now, I only have proof that it works in vitro. So what we propose on doing is, instead of using embryonic neurons, we will use adult neural cells. So adult astrocyte, adult neurons, different ages, okay? Not only in terms of age, but I will show you there's also some sex-dependent aspect that's very important, and I think John will talk about that as well tomorrow. Moving forward in the preclinical aspect, we really need to understand that the same drug works the same way or different way in younger animal or older female, for example, versus older male. And finally, based on this data, perhaps we should, when we move in clinical trial, let's say you have a drug that was specifically or works 2 or 3 times better in young female in vivo, why should we test them in other males in clinical trial? Maybe we should focus on that right population that we have in preclinical to demonstrate it works. And then if it works, maybe extend it to humans in older ages or sexes. So to do that, the first thing we have to do is develop an in vitro system, because there was no, until like 2 or 3 years ago, it was impossible to actually culture adult neurons from different ages, cortical neurons in vitro. And so I will be quick, and we published that very recently. We developed this system based on mice where we can separate the glial cells from the cortex, from the neurons, and we're using this robotic system where we can actually screen for 10 to 20 drugs at the same time and quantify the neurital growth assay and quantify the cell survival. And I'm just going to show you 3 different drugs to demonstrate what I was talking about. So it doesn't matter what drugs do and what the molecule targets. The point is, for example, the drug A can enhance growth independently of age, all right? So if you have a 9 weeks old cortical neurons or 27, this drug A in blue enhance growth the same way. Drug B, and that's an example that if you use, depending on age, you may enhance growth or actually reduce it. So this is it's kind of a drug that can be detrimental if you move it forward in preclinical trial in middle-aged animals. And drug C, an example that actually in embryonic neurons, I didn't put it here, but it was reducing axon growth in embryonic neurons. It was not having any effect on the 7 weeks old, and it was only positive in these 27 weeks old cortical neurons. So this is the age component, but there's also a sex component. This is the same drug A here, where we did different concentration, comparing the old female at the bottom. In blue here is the male, the young male, and red here, the young female. So clearly, you can see that the same drug have almost 2.5-fold more efficiency in the young female, at least in vitro, than it has in the young male and the older female. So the next thing we wanted to assess is what's happening in astrocyte, because astrogliosis modulation of astrocyte can be involved in inflammation and synaptic plasticity and axon growth. So using very similar system and similar mouse, we developed a scratch assay where our basic idea for acute injury is to actually to speed the wound healing, to very close it very quickly. And hopefully by doing so, we will enhance neuroprotection and reduce the loss of locomotion. So this is an assay that we developed in the lab. We also developed like an in vitro model for a more chronic stage, if you wish, where we put different drugs to activate the astrocyte. We can also use a scratch assay and have a kind of subchronic aspect where we test drugs and we're trying to see if we can reduce inflammation, reduce axon growth, modulate the astrocyte to enhance axon growth and enhance synaptic plasticity. And again, I just want to show you one quick example of two drugs. So the blue one here at different ages on astrocyte, and this is the scratch assay. And so this blue drug actually promotes cell proliferation of the astrocyte and reduce the lesion, the injury size. And that seems to be depending on aging. However, the red one, it completely the reverse. If you use the red one, it seems that you increase the size of the injury, which is not something that we want. And so the next thing I want to mention is, so it's working in mice and we can do it in rats. But the thing is, we can only screen for a certain number of compound at the same time. We can't go maximum, we can go like maybe 20, 25 compound maximum. So that's why we wanted to develop a new system to really have a high content screen assay. And for that, we moved for a mouse, from a mouse to a sheep model. And we're using 2 or 3 year old sheep, and we developed a completely new system to purify the neural cells. It's a fast process, and it's much cheaper than using the mice. And we can have millions of cells that we can test for thousands of compounds. And I'm just going to show you a quick example. So right now, the way we do the screen is in these 4 steps. So the first step is using this 2 year old sheep, where we screen thousands of compounds. And for say we have 30 compounds that we move in step 2, where instead of using 2 or 3 month mice, we will use 6-month-old male and female, all right? Adult cortical neurons. Now, once we have these dual species positive hits, we will move and validate and try different concentration in both 6-month and 18-month male and female. And only then, hopefully, we'll have this dual species, age and sex drug that will work independently of age and sex that we will move in vivo in, again, 6-month and 18-month in male and female. And so so we're starting doing this kind of work, and we kind of made a specific compound library of 12,000 compounds. And using in sheep model, from this 1,200, we have 150 that were good enough for us to potentially move forward into these different steps. And these are just examples of drugs that we found from our screen model that have either been used to be beneficial in preclinical model in rats or mice, or are different phases in neurological disorder in humans. So showing, at least to us, that these screen steps can identify drugs that could be moved along the path. So right now we're only focusing on drugs that never been used in human neurological disorders. So we have these 30 compounds. We validated all the way down here. We have 6 compounds, and now we're starting testing in vivo these 3 compounds. I don't know what's going to happen. I'm just telling you this, the way we're doing it and the way we hope it would enhance the chance of success. And again, the drug that we move forward are the drugs that are positive in sheep and mice, male and female, and young and aged animal neurons, okay? That said, we might as well, in the future, focus on one particular drug that work only in young female or older males, okay? Right now, we're only focusing on the one that work independently of age and sex. And so in summary of the second part, I hope that I've shown you that we develop a new system for adult and astrocyte culture that we can use in mice and sheep, that we can test different drugs. And again, the idea is to work the best way. Of course, we find a drug that works independently of age and sex, but perhaps we have to rethink about that and move along this kind of group personalized medicine, where if you're an older male, we will give you a drug A. If you're a younger female, we will give you a drug B. So in conclusion, again, that's something I said. So if you're 20 years old, male or female, or 60 years old at the time of injury, the spinal cord injury will respond differently. And if you wait just even 5 years, and it's even worse if you wait maybe 10 years, the injury will be different than when you were 5 years ago. And we need to understand what's happening, and that's something we're trying to do in my lab, at least in rodent. We're trying to understand the molecular mechanism acutely and chronically, and how this different drug that we're testing can actually be applied to all these different groups. So perhaps in conclusion, and that's something maybe we can talk more like in the debate in the future, but I really think that if we, at the drug discovery stage, if we take age and sex into account, we will have increasing the chances of having a drug moving forward and be efficient in the preclinical stage. Now, if we go to the preclinical, I really think we have to use age and sex groups matching what's happening in clinics. And based on that, I really think that we have to refine the way we move forward in clinical stages based on the data that we have in vitro and preclinical, and hopefully that will enhance the success rate of any potential strategy. And with that, I just want to stop here. And if you know anyone looking for a postdoc to work on this kind of screen system, just let me know. And thank you.
Thank you, Cedric. Candace, come on up. Now you're going to hear a little different story. As I mentioned earlier, Candace works with pigs in a pig model. She's going to tell you all about that. But I also just want to emphasize, I really appreciate Candace in particular for what she commented on earlier, that she has made her lab available as like a translational workshop, kind of, to collaborate with as many folks as she can. And I'm really grateful that she collaborated with us when we approached Candace and Jerry and Anne and NerveGen to do this combinatorial study, that they were all willing and excited to do it. And Candace is where the home of the project is. So grateful for that. And she's going to mention that also. Okay, welcome.
Okay, so this must be Cedric's computer. I'm not going to give his talk. Just make sure I couldn't do that as eloquently. Okay, well, I'm just going to ask you to advance the slides. Tell me. You're making signals to me I don't understand. I'll be like, I can do interpretive dance. You should just tell me. Is this the right one? Okay, perfect. I was gonna say, is it the big green button? Perfect. So thank you all for coming. I've already seen people that are some of my favorite people in the world here. And I just reconnected with one of my favorite friends from 20 years ago. So I'm already having a wonderful conference. I've learned a lot and I've already been inspired. So what I am tasked to talk about today are some of the key variables that are likely to affect translation. So I thought we would take a step back and I would talk about some terminology and what I'm referring to when I talk about translation. So what I'm referring to is this idea of taking something. You've seen wheels, you've seen a gradient that Cedric shows you, something that takes an idea, from the early stages of an idea through the research spectrum to clinical practice, to use, right? That's what we think about of translation. So the NIH's definition for translation is a process by which discoveries are evaluated in different systems and different applications. So the ultimate goal of scientific discovery in spinal cord injury, development of new spinal cord injury therapies, is translation. So whether you call that cure, whether you call that regeneration, whether you call that repair, that's the goal. So about gosh, I guess I was supposed to tell you what I look like I forgot this. So I'm not going to tell you how much I weigh. I'm going to lie. And I'm going to tell you that I'm 5'5" and I weigh 120 pounds, which is an absolute lie. I weigh a lot more than that. I have red hair. I dye it, but I was born with red hair. So that will explain a lot of my bad behavior. I'm probably among the most tattooed people in the room, but I'm wearing long sleeves. So no one knows that yet. And other aspects about my personal appearance we'll leave to the end for interpretation. So let's talk about these key variables again and how these key variables affect translation. So what I started to say before I lied about my weight, I'll lie about my age too in just a second, but before I lied about my weight was that about 10 years ago, and Susan is in the room, she's one of the people that inspired me to make a change in the laboratory. So I decided to make a conscious decision to change the way I was running my lab from it being that one shop cottage industry, the way we were trained, and being that competitive lab that competed against everyone else in the room, to be focused on developing translational tools that everyone could use. And I'm going to talk to you about those today. And in that focus of revamping the lab to have shared collaborative translational tools, we decided that there were key variables that needed to be considered. And species and sex are the two that I'm going to talk to you about today. So I also want to talk to you about the term sex. So in this context, this is not Game of Thrones sex. This is a different kind of sex. This is sex as a biological variable. And I think that Matthew said gender. So the difference to a biologist between sex and gender is sex is the anatomical, physiological components that the animal or the person is born with. So it is gonadal hormones, it is anatomy, those structural components. Gender is the way that we as humans interpret our role of those components and our role in society. So when we're thinking of sex as a biological variable, it's actually sex and those physiological components that underpin our interpretation of gender. So just to clarify that term. So species and sex are the two things we're going to talk about. Age, diet, resilience, care, and housing are all really important and things that we are interested in the lab and have worked on for some time, but I don't have time to talk about today. Okay, so you know this already, but I wanted to give you a little bit of a backdrop for why, again, about 10 years ago, I decided we were going to change the way we did things. We had that idea that if we continue to do things the way they've been done, we'll continue to get what we already have. And that was not acceptable. So you know this already. So this is showing success rates for CNS drugs. And I've drawn a few little elephants in the room, but what you can see is that the amount of time it takes from FDA data for CNS drug development on average is it takes about 35 years from initial idea to clinical trial. And the success rate for drugs in all of the CNS space, so this is not spinal cord injury drugs alone, this is all drugs in CNS, Alzheimer's, Parkinson's, all different types of drugs. The success rate is about 5%. So to me, that's unacceptable. That the current discovery process is time, labor intensive, and failure ridden. So there is an international, interdisciplinary, active discussion of which you are all members of that discussion to improve this. And I have dedicated my life and career to improving this. And so our contribution, as I mentioned, is kind of this idea of a pipeline. So the goal is for us to develop novel tools that all researchers could use that will advance research along this research course that's been discussed. So the terms, they're basic research, translational research, clinical research. They're used at different stages and they mean different things. But what I'm going to talk about are the stages that are prior clinical research. And I'm going to call all of that basic and translational research preclinical. So our overarching idea in the laboratory is that if we improve the models, if we improve the rigor, if we improve the reproducibility of what we're doing, If we are transparent about this, if we share ideas, if we include everyone's perspective, this will accelerate discovery. And you know what? I don't really give a fly and flip whose name is on the top. Don't tell my chair that, right? Thankfully he left earlier, right? Don't tell the advancement committee in my institution that. I'm old, I'm advanced already, I'm good. What I want to do is advance the new cures and new treatments. And if someone else gets credit for some of that work, I think that's fine. Okay, so let's talk about species. So we've already kind of talked about it a little bit, but I wanted to make sure you understand that the work I'm going to talk about is work in pigs, and that the research that is conducted in these pig models is conducted with compassion, it's conducted with integrity, it's conducted with intellect. We work hard in the lab, we have fun in the lab, we respect the animals, we respect the question, but our goal is to translate the research. So it's highly regulated, federally regulated research, and we are rigorous in all of our adherence to federal regulations. So why pigs? Out of all the things, so Cedric told you he started working with sheep brains, and I'm excited, and we're developing collaboration. But pigs in biomedicine are actually really amazing tools. So pigs are more similar to human than rodents in many regards. Anatomy, complexity of the brain and spinal cord system. Pigs are closer to human than many non-human primates. The cardiovascular in pigs is so close to that of humans that pig tissue is used in replacement surgeries for human cardiovascular surgeries in some instances. The gastrointestinal tract of pigs is identical. I don't have time to talk about it today, but we fed pigs a total Western diet to get metabolic disease, and we were able to get that. There's a certain breed of pigs, they're called Osabel pigs. They developed an island off the coast of Georgia that developed metabolic syndrome identical to that of humans. So pigs are very good models for types of things that affect human medicine. Pharmacokinetics and pharmacodynamics are terms which explain drug delivery and how drugs affect a system. They're almost identical in pigs to humans, right? So you cannot give one example of that is you cannot give a mouse or a rat enough nicotine to mimic the nicotine processes in humans. But for pigs, it's very similar. Just to give you an example of a commonly used drug. The genome of the pig has been sequenced, and it's 90% similar to humans. And again, as I mentioned, pigs get fat, especially if you feed them high-fat diets. They get obese and develop metabolic disease. Pigs have already been successfully used in many areas of research, including human coronary artery disease, infectious disease, toxicology testing, vaccine development. Skin from pigs is almost identical to human skin. And even in burn victims, a lot of victims will receive pig skin as implants. There are many models of pigs of human disease. And as I mentioned, there are cancer models and tumor models. So pigs are actually very good predictors of human disease. In terms of neurotrauma and spinal cord injury, they have a complex physiology and a complex anatomy that mimics that of the human. And I'll talk more about that. This is a paper from my lab where we ask a very simple question. The cortical spinal tract, the main descending motor tract that controls restoration of movement and is very important for restoration of function, is where is the pig cortical spinal tract? Is it anatomically similar or dissimilar to that of rodents and humans? So we used image-guided neurosurgical techniques right out of the clinic. We literally rolled over some equipment from the OR. We did this on Sunday when they weren't using it. We cleaned it off and took it back Monday. So we use the exact same equipment that's used in humans and pigs to do image-guided neurosurgery. We use that today in quite a lot of experiments. And just a figure from the results of the paper show, the top panels labeled D, E, and F just show the tracing of the axons in the pig spinal cord after a fluorescently injected tracer. On the right of that is the location of the rodent cortical spinal tract. And then what you can see immediately below that, I think I have a pointer. Yeah, immediately below that is a human anatomical reference of the location of the cortical spinal tract. Here it is in rat. Here it is in human, and in pig, it mimics the location of that in human. So just an example of how in the anatomy of pig mimics that of human in the cortical spinal tract. There are lots of challenges to inducing spinal cord injury in pigs. Pigs get pressure sores. Pigs have neurogenic bowel and bladder that we have learned to manage. The paralysis in pigs is much longer lasting than it is in rats, and I'll show you some data on that. Another thing, we talked about money earlier, and I'll talk about money here. So it costs me to do a study in one pig, acute study, 7 days, about $20,000 per pig. You can do an entire rat study for $20,000 in most instances, and that's an acute study. The study that we're talking about that Matthew referred to earlier is a chronic study, which we can also do in pigs. So much more expensive study. So there is an opportunity, and I'll talk about this, for people to collaborate with my laboratory. So the good thing is we have spent the last 10 years setting up pig models, setting up a neurocritical ICU, all the care units, all the monitoring, back translating outcome measures from human clinical practice to the pigs. I won't talk about it today because I don't have time, but I speak pig. I took a sabbatical at Cornell University to learn bioacoustics. We have mapped the vocal repertoire of pigs so that I understand pig speak. Pigs are, this from movies, but pigs squeal and talk and vocalize 100% in our audible range, and we can classify that. So we understand the pig's interpretation of what's going on in their life. So let's talk about sex. So this is probably something you'll also see from Dr. Genzel. John Genzel's a collaborator of ours, and this is from a great paper that his team worked on. And what they showed in this paper is they were looking at rodent studies, and they were looking at studies that were from preclinical rodent studies funded by the National Institute of Health. And the red pie is females, the number of studies that used only females. I don't know where my time is, so is that the green light there? Okay. The red study is only female. Now, the NISCUS data suggests or indicates that about 78% of new cases in humans each year are male. I don't have my glasses on, but someone tell me, does that say 71.6% of NIH-funded studies in rodents are done in females? Right? So a big difference. Now I see the timer. Now I'm rolling. Now I know where I am. Right, so the idea here is there's an incongruity of sex studied in preclinical studies. So there's something interesting about the pig. So the pig has an estrous cycle that is more similar to human than in rat. And I don't have time because I talked too slowly in the beginning to go through this, but this box here shows the rat estrous cycle. That cycle lasts about 3 to 4 days. It's very peaked, it's very short, it's very sharp. The pig estrous cycle is shown here, lasts about 25 to 28 days. And the human estrous cycle, so this is human, sorry, and this is our data from pig. And you don't have to be, looking at this quantitatively to say that these two look more alike than this one. So the pig is a very good model for studying sex and hormonal differences over cycle than human. So I'll go through this a little bit quickly. But we did a study, and our question was very simply, are there differences in recovery after spinal cord injury between male pigs, gonad intact male pigs? We're one of the only groups in the world that studies gonad intact male pigs with spinal cord injury, and female pigs injured at different stages of the estrous cycle. So the answer is yes, there are differences. So there are differences in the acute pathophysiology. So this is looking at gray matter sparing at the epicenter of spinal cord injury. Uninjured controls that receive the laminectomy only are here. And this is looking at number of neurons in the epicenter of the ventral horn. And what you can see is the males at this early time point have more preservation of tissue. What's a side note is that I studied this same phenomenon in rats for about 15 years before this, and this is opposite of what I found in rats, where I found that females had more spared tissue. The same trend is seen with white matter in the early time point, more spared white matter in the males than either two of the female groups. And at the later time point, 6 weeks after injury, that pretty much evens out. So what this suggests, and same with white matter. So what these data suggest to us is that there are sex-dependent differences in the pathophysiology of injury between male and female pigs. Now you ask the question, what about the function? And there are a couple points I'll make here. So this, oops, someone threw away. Okay, this graph depicts the functional recovery. So this is a lot like those Bezo-Beatty-Bresnahan graphs you're used to seeing in rodents. Uninjured controls are here. So this is normal walking. Stepping is at the red line. The first thing you should notice is that none of the pigs step and none of the pigs recover stepping after this injury model, right? So it's a very clinically relevant recovery. Looks a lot like a human recovery. And that you also see that there is a non-significant trend for the males to recover better than the females. So again, there are some sex differences in the pig model. So what's our approach? I'm on time, a minute. So our approach then is to incorporate these clinically relevant variables into a research design. And I talked about this pipeline already, so I want to talk about a few things. So we are here and available to collaborate. We have all the tools set up to do everything from neurosurgery to pressure sore management, bowel and bladder management in these pig models. So we welcome discovery, as Cedric gave you this beautiful model for discovery of new drugs and compounds. And then we can do scale up and validation. I am a good laboratory practice professional. That is the standard of documentation that is required for FDA submissions for investigational new drugs. So we can eliminate time and steps by combining these models, and that will eventually take us to further steps in the path. So I wanted to give you a couple of examples. I mentioned one earlier, but I want to give you a couple examples of how this pipeline is working. So I'm excited to have a collaboration with Jackie Sagan from the Miami Project. Jackie spent about 20 to 30 years working on a gene therapy approach for neuropathic pain after spinal cord injury in rodent. Didn't have anywhere to take it, brought it to us. We're collaborating on this wonderful project to scale up and test this novel therapy. I know I'm out of time, I'm finishing. As I mentioned earlier, I think Joanna is on by virtual, but from this meeting came a collaboration with Joanna Stanica from Exonus. We now have a it starts October 1st, so in a couple of days, a newly funded project to take a KCC2 potentiation therapeutic that they had developed and tested in rats, scaling it up, testing it in pigs, getting it ready for FDA. John Ginsel will talk about our evaluation of sex differences in immune responses and a continuing ongoing collaboration. Really exciting work there. The collaboration we talked about that many of you here are supporting is with Ann Parr, Jerry Silva, NerveGen Pharma, who are also here today. And that project is to test a combination therapy for chronic SCI. So these are all examples where I say we have got the tools. I have the professionally trained staff. Many of my staff members have been with me for over decades. We have the team that is ready to do these experiments. Bring your ideas, bring your combination therapies. We will test it in a rigorous, robust way and help accelerate the field. So what are the take-home messages? Porcine models are not perfect. Quinn will tell you that we worked on this project and a porcine model and we hit a roadblock with that particular drug. There are times when they don't work, but they are more human-like. They have an anatomy, they have a recovery profile, they have pharmacokinetics and pharmacodynamics that are nearly human-like. They are very good models of humans, but let's face it, folks, we're not pigs, right? We have differences that are important to understand too. My lab has developed many tools for collaboration. We speak pig, we study sensory function, we study motor function, we study bowel and bladder function in pig, pressure sores, all of these things that are important to not only advancing care, but advancing cures in the spinal cord community are available, ready for collaboration. And I think that sex differences, sex and age, as you've already learned in this session today, are very important variables for consideration. And we, again, are one of the only labs in the world that does spinal cord injury in gonad intact male pigs. And we have developed specialized tools to do so. I can tell you, I have been knocked on my, you know what, by a pig many times that's headbutted me or challenged me or run me over. We're not talking about a pig. I'm talking about a pig, right? So these are large animals and very opinionated sometimes animals, but we have the ability to handle them and advance research. So I thank you very much for your time and your consideration today, and I'm excited to have a further discussion.
Thanks, Candace. Yep, thanks, Cedric.
Teaching evaluations, they always say I talk too fast and my hair is distracting. So I'm sorry.
Questions? And wait for the microphone, and please introduce yourself.
Hello.
Yep.
Okay. I'm Dave Gillespie. I'm actually a researcher at Huntsman Cancer Institute here in at the University of Utah. And we use rodent models specifically for our work. And that's been one of my biggest criticisms of spinal cord injury research is that they focus on rodents, that the rodent CNS is completely different than a primates. You know, it's completely different than a humans. So I applaud you for transitioning over to pig, and I'm wondering why I never heard about your lab. Because the question is, do you think that pigs are going to that we can rely on pig data without having to do any primates before human trials?
Thank you for the question. I have a couple of suggestions there. So one of the main differences between the spinal cord injury that we induce in the pig models currently and spinal cord injury that's induced in many of the primate models, and there are collaborators here in the room that work with those models, is that we do a clinically relevant lesion. So we do a contusion compression lesion that results in, as you saw, a high degree of functional paralysis. So as I mentioned too, I have protocols in place to manage neurogenic bowel, neurogenic bladder in the pigs, to manage pressure sores and other things. So there are clinical complications that come with that severity of injury in the pig. And I think that's important. And so I can't tell you how many times in the middle, you probably never heard of me because I'm in the OR in the middle of the night, right? You know, doing something that has to be done to manage these animals, medically manage these animals. And so I think that there are value in the primate models as well, but typically the primate models in spinal cord injury use smaller lesions that incur less impairment in the animals for lots of reasons that many of them you know. As far as are pigs going to be an acceptable replacement for primate models when we're talking about FDA approvals and other things, we have had success in that area. And so the FDA, in my experience, and it's only my experience, has been willing to accept a well-justified model, and it does not have to be an NHP model. So as long as it is a well-justified, anatomically relevant, clinically relevant injury, the FDA and these applications we've been working on under good laboratory practice have been very well received. So I do think it is a viable alternative in certain instances for NHP models.
Can I ask Cedric, is increasing the variability in a rodent model an adequate substitute?
In vivo? In vivo?
Yeah.
So I think it's very complementary because I don't know how many drugs you can test at once, but you know, with rodent, you can test several, and right now we're testing 4 at the same time at different concentrations. And I don't think you can do that with the pig or monkey model. So I know, to me, for speed and the way I would see it, I would go in the rodent, in the mice or the rat, and then take the best one with best concentration to move forward with the monkey or the pig. That's how we do it. And we use the same T8 contusion compression, so it's still very relevant to clinical translational. It also depends on the question you want to address. If you're looking like the model I showed to looking at sprouting, this is not something that occurs in spinal cord injury patients. It's just a way to see if we can enhance specifically sprouting. If you look for a contusion compression, then it's more clinically relevant.
Question?
Yeah. Hi, Marco Baptista. I'm from the Christopher and Dana Reed Foundation. I want to commend you for, controlling for those factors in the preclinical space that a lot of times people don't address. But I had just a general question. When you're screening for the drugs or repurposed drugs, do you think there's a challenge in the field in making sure that people gain access to the full characterization of those drugs, for example, selectivity or any other data that might be in an investigator's brochure prior to getting too far along in the drug discovery process?
I don't know much about all this PK and the translational. I'm not really qualified to comment on that. What I would say that, for example, the FDA and we trying to repurpose the drugs in terms of grant funding, it's very hard because the grant people granting the reviewer, they say, well, it's a drug that's been used for A, B, and C, which we try now to repurpose it for spinal cord injury. And so far, I didn't have any success in doing that. So I don't know if that's something we should look into and discuss, but yeah, I can't comment on the other question.
I think it depends on the nature of the collaboration. And so I think that in my instance, and partially I think it's because we also work in the GLP space. So we have been collaborating with several industry partners and, under non-disclosure agreements and other things. And so I think it just depends a little bit on, how the team sees that moving forward. Yeah.
Linda. Oh, is there someone else first? Go ahead. Monica.
I'm Monica Perez from Shirley Ryan Ability Lab. So I have a question for Candace. So thank you very much, Candace, for the nice presentation. But I have a question about the corticospinal tract, because as you mentioned, is one of the most important descending motor pathways for recovery. However, in humans and in primates, one of the important aspects of this descending motor pathway is that we have direct projections to spinal motor neurons, and this is why they contribute specifically to skill, movements, and dexterity. Do pigs have direct corticomotor neuronal projections? How different is that systems in that model?
Yeah, it's a great question. And I applaud you for asking it because it's a very good question. So to the best extent of my knowledge, that level of anatomical detail that has been classified in the non-human primates, in cat models, for years, and in rodent models is not as well known for the pig. So when we did our corticospinal tract tracing paper, that was one of the earliest papers that started to kind of look at these types of anatomical connections in pig. We used image-guided neurosurgery to inject only a small location in the motor cortex. And we have not done, although we have ongoing projects to work on doing things like tracing the rubrospinal tract, looking at polysynaptic connections, looking at other aspects of motor spinal tract. So that type of work really needs to be done. And it needs to be done, I think, to really advance some of these questions that we're talking about using the pig as more of a discovery model, and to understand the synaptic connectivity better in the pig is really an important next step.
Hang on, Linda. I just want to there's a question from the live stream. If you're ready. Well, let's just do one and then we'll go to Linda if we have time.
So the first one is from Corinne Jean Mayer, and this is to Cedric regarding regeneration and aging. Do you know if the age-dependent regeneration decline means that fewer axons regrow, or could it mean that they regrow much slower? In other words, provided axonal regrowth is duly promoted, is the growth speed 1 millimeter per day, as known in young age, still valid in an older central nervous system?
Yeah, it's both. Yeah, we know they regrow much slower too. So if you, for example, the P10 story that I'm presenting, if you wait 7 or 8 months, you will still see some axon regenerating. So it's both in number, in terms of speed. They have different intrinsic that we're looking into. I don't want to go into the detail, but yeah, it both ways.
Linda.
Linda Jones here on behalf of the Better?
Yes.
Linda Jones here on behalf of the American Spinal Injury Association. So Candace, you touched on this a little bit in terms of that you use a single drug at any given time. Are there other types of investigational preclinical studies that are not great in the pig model? Can you comment on that?
Yeah, so I think that the challenge, there are a couple of main challenges that the pig model has. So, and I think it gets a little bit back to NHP question. So there's certainly quadrupeds, right? So there's that. So when you think about loading along the spinal column, the loading in a quadruped is going to be different from a biped, certainly. There are also ungulates, right? So no digits, right? So fine motor function, they have hooves, right? So fine motor function, those types of things are going to be better studied in animal models with, prehensile digits. So absolutely, those are limitations. I think that the choice of the model is really important to advancing the translation, which is probably what you're getting at here, right? Is that it's certainly not a one size fits all. In the experiment that we talked about, it was I'm trying to figure out how much I can disclose without violating the NDA, but it was a protein-based product. And it turned out that the pig had a very different receptor profile for that particular protein than the human or the NHP. So there are, instances like that where, it's like we were talking about something that hasn't been investigated in the pig, and there's no reason to investigate that protein product in pig previously, because why would you or whatever? And then we discovered that there was toxicity. I think there are some advantages too, because pigs essentially, thinking about gene therapy, because pigs essentially live in the same environment as humans. So, endogenous AAV inactivation proteins, for example, are very similar in pig and human. And so we had a grant that went in before, and they said, well, that's a disadvantage, right? Because the pigs have these similar AAV inactivating mechanisms. And we said, well, actually, that might be a disadvantage that we can turn into an advantage, because the humans will have the same inactivating mechanism. So I think it depends quite a bit on the question, and the outcome measures that are being evaluated need to be carefully considered to select the best model. So again, a pig is not a perfect model. It is a model. It has limitations for sure. And I think it also has potential, but thank you for highlighting that point.
Another live stream question.
Okay, so this is from John Chernesky, and he asks, given the very high cost of the pig studies, would you advise to first try with rodents and then go to pigs, or skip rodents altogether?
No, absolutely. I mean, it's what Cedric and I were saying. So the pig studies are extremely high cost, extremely labor intensive. So we see them as a translational intermediary. So I think there needs to be robust development, as Cedric has shown you, in rodent models that have careful consideration of key biological variables, use the ARRIVE standards for reporting, are open and transparent, and let's then take the best and most robust strategies from those species and move those up the phylogenetic tree. That's essentially the idea of the pipeline, develop the tools to do so.
And I would add that the price, like how much it costs to, I don't know if you have a small 30-gram mouse or 30 kilo, how much drug you have to put in the one pig. So if it's expensive to produce, it will be really hard to really test it.
Yeah, that's actually, I've about 100 funny stories that require beer, but where I've been to a pharmacological company, like a small startup company, and they say, we're ready, and then I talk to one part of the office, and then I go to the formulation group of the office, and they say, how much drug do you need? And I tell them, and they're like, there's no way we can make that much drug, right? So those, considerations on scale are really important at all stages.
Okay, so he has one more part to that question where he says he's also like to hear Cedric's thoughts on small animal autonomic measures.
The other side of what I didn't talk about. That's another 2 hours talk. So specifically, so we're looking into bowel dysfunction and how we can do electrical stimulation of the bowel. So, and that's why we want to collaborate with on the pig model. We're looking into cardiometabolic disorder and finding drug that can reduce liver fibrosis or this kind of aspect. But I mean, it's a never-ending open question. I don't know how I can answer that, but just email me or I don't know.
Do we have another live stream question? No, we don't. It's time for lunch.
Thank you.
We're going to stop. Thank you, Candace. Thank you, Cedric. Appreciate it.